Devil's facial tumour disease, DFTD, is an aggressive, non-viral, transmissible parasitic cancer which affects Tasmanian devils. It is one of only 3 transmissible cancers in the world. The first occurrence of DFTD was observed in 1996 and has since had devastating effects on the devil population. Since its first appearance, the population has fallen drastically and decreased by 70%. Studies predict that 80% of devils are currently infected, and they have been endangered since 2008. The disease is characterized by the appearance of obvious tumours that appear around the mouth and face of the animal. The first visible signs of the disease are lesions or small lumps that form around the mouth and face. They originate within the dermis or the subconnective tissue of the mouth. These then develop into large tumours that are typically concentrated around the neck and face area, but in some cases may also be present on other parts of the body. The facial tumours make it difficult for the animal to ingest food, and whilst death can occur by the breakdown of internal body functions due to the cancer, it is usually a result of starvation. The affected animal becomes progressively weaker, and as a carnivore is unable to catch and consume prey. However, one study reported 73% of cases showing signs of necrosis. Invasion of the lymphocytes, vascular tissues and heart was also seen, but only in a small number of cases. After the appearance of the first tumour, the infected devil usually has around 5 or 6 months before death occurs. The cancer is spread when the devils bite, which occurs often as this behaviour is incorporated into all aspects of their life. They fight when trying to find a prospective mate, protecting territory, through social interactions, for example play fighting, and when around food due to a hierarchical system within their group. The cancer can also be spread if the devils eat a carcass of an infected devil or through the sharing of food with a devil who is themselves infected. This can result in unrelated devils developing the cancer and increased transmission through separated groups of devils. The cancer is also spread more easy due to a low genetic diversity and a chromosomal mutation unique amongst carnivorous mammals. One way to stop the spread is to quarantine infected individuals. Clinical diagnosis involves examination of the growths to see whether they present as solid soft tissue growths that are usually flattened and ulcerated. A biopsy of the growths will confirm DFTD if tissue samples present round and spindle-shaped cells when treated with H&E staining. By the time DFTD is diagnosed and distinguished between different types of neoplasia, the disease is already in a well-developed stage, which may or not be metastasizing. Preclinical diagnosis is an essential part of the conservation of the species by isolating infected devils, hence reducing transmission. Research such as testing for biomarkers and identification of specific peptides are still in its infancy. The most promising biomarker for DFTD is periaxin, but these preclinical diagnostics are only viable in controlled environments that do not require large-scale screening. The cancer arose from a mutation in a single female Tasmanian devil, nicknamed the Immortal Devil, due to the cancer mutations living on and being present in all the future infected devils. Siddle and Kaufman found that the immune system in present devils does not respond to the cancerous cells. They found that there are no MHC molecules present on the cancer cells. These molecules are usually associated with the cancer cells, which are recognized by the immune system. Their later work indicated that due to the Immortal Devil's mutation, which arose in the cancer, the gene expression for these molecules had been turned off. They now believe that there is a possibility to turn these genes back on in order to express MHC molecules and initiate an immune response from the host. The idea is to vaccinate the devils by injecting cancerous cells with the MHC molecules already attached, and hence the cancerous cells would be recognized and attacked by the immune system. However, there are many risks in proceeding with this proposed treatment. Using the few remaining devils left is controversial and could potentially increase the rate of extinction of these animals if proven ineffective. It may also be too late to vaccinate the scarce few devils left. Perhaps breeding programs are the only solution for these critically endangered species.